I will include human mutations. How can we categorize human mutations into major divisions? There is a reason for categorization, right? So let's divide them. What about chromosomes? Chromosomal abnormalities result in chromosomal anomalies. Mutations? I'll simplify it a bit. What other terms are there? Genetic abnormalities, mutations mentioned earlier. You may have seen questions on the college entrance exam, but these concepts are not covered there. They are for internal assessment. Actually, so anyway, once we cover the concepts and move on, we express that there is always a basis for division. Why did we divide them like this? Since there's no answer, I guess I'll have to figure it out myself. Got it. The criterion is karyotype analysis. Can we differentiate them through karyotype analysis? Yes. Karyotype analysis is not possible. Let's divide them again like this. The detailed information will be covered in the next part, so wait for it. And mutations can be broadly categorized. We learned about structure and number. First, there are some notes on structure because of structural abnormalities and mutation. We can probably identify chromosomal structure abnormalities through karyotype analysis. Then there are numerical abnormalities of chromosomes, chromosomal numerical abnormalities, and structural abnormalities and mutations, and in detail how the structure is fragmented, duplicated, inverted, and translocated. So we'll cover these four aspects. Fragmented structure, check the details later. Then, for inverted structure, it's inverted here. And for translocated, it's translocated between non-homologous chromosomes. That's how we'll learn these four aspects. Concepts are very simply not covered much in the college entrance exam. They are for internal assessment. For internal assessment, and then numerical abnormalities of chromosomes, chromosomal numerical abnormalities. Mutations will be separated into two categories. How are numerical abnormalities caused? And of course, what causes structural abnormalities? Today we can leave comfortably in just 30 minutes. This non-disjunction is already done. If it wasn't, I would have had to order a lot. But because of the abnormal non-disjunction, how does the autosome from the sex chromosome caused by autosome non-disjunction? There must be some non-disjunction. Let's write it down first for you guys too. And then let's fill in the rest with what we already know. Okay? Through sex chromosome non-disjunction, who is created by chromosome non-disjunction? Guys, we're going to learn exactly two things that come from this. We're going to learn about one person, Down syndrome, and then Edwards syndrome. Like this, it often appears on the test. It's Down, but we're going to learn about Edwards syndrome like this. So we learned about this like this. This is how the number of chromosomes is. What's abnormal about Down syndrome is the 21st micromole, the chromosome is 3. This one is the 18th chromosome is 3. So when we express this in chromosomes, we express it like this, how it's detailed on the back. I'll just do it and move on how, guys. Originally, it's 45 plus woman is XX. Man is 45 plus XY. This is also the same. Instead, this is where the 18th chromosome goes in like this. Do you understand what am I saying, what I'm expressing? Like this is at the back, because I won't say it. In the back, there's all the sex chromosome non-disjunction we studied earlier, right? What we learned? Our sex chromosome. Since there's a lot of this these days, I'll write down the number. Shall we start from Turner? We'll start from Turner. Turner, then Klinefelter, this. Here and there, in Bundang, there were exam questions. A student wrote Klinefelter in the descriptive question and told me. Suddenly it came and our teacher said, I don't know either. He wrote Klinefelter, the teacher would probably say it's correct. I don't know either. Anyway, what should we do next? Superwoman, perhaps? It's okay to call a superwoman super. And then there's something like supermale for the fourth time. You would know this, right? Basically, the basic chromosome structure is 44. Because it's a secret of gender, there would be 44 of these all the same from the state where there are all 44. Looking at the chromosome composition, there's only 1 plus X. So it becomes 45. You know, Turner is now plus XXY. This is a man, and this is a woman, XXX sometimes. There's a person who asked a question like this, teacher this. What happens if there's only one Y? This won't be born. So it's not treated as a disease. Don't check this, okay? I'll wrap up next. Memorize this below too. The teacher already made you memorize it. Last time's test question. When solving, I solved a lot of problems at the beginning. We did the part where there's nothing you can know through karyotype analysis, right? Genetic abnormalities and mutations, compare this with this. That's how it always goes, appearing in the front part of the test, right? Among the next, what can't be known through karyotype analysis, appearing like that. In fact, color blindness is also a genetic mutation, guys. But that is an example. It doesn't come out well. What always comes out well in the test paper? What goes well? They say 5 things go well, and you have to memorize those 5 things. We experienced everything while solving the problems in the front, right? You don't have to show the front. The problem is, these are 5 things. This is cat cry syndrome, our cat, you know, a pet cat, right? So, cat cry syndrome. Wait, I made a mistake. No, it's changed. Wait, cat cry syndrome is actually a miscarriage. So even if this fits well on this side, I get confused from the cat's cry. This goes well in the multiple choice questions, structural mutation. Sorry, I made a mistake. I was fooled. There are people who memorized again, right? Cystic or cystic, going out at the very end. So cystic fibrosis. Cystic is also fine. Then again, albinism. 4 years ago, it was suddenly changed to leukoderma. It was changed to albinism because we decided to use our language like North Korean. It was perfectly changed to leukoderma, then it came back again. It turns white due to albinism. All the pigments are lacking, so they die quickly. Albinism, phenylketonuria, also sickle-shaped red blood cells. Anemia, originally, red blood cells should be beautiful in a round shape. In the end, in the sickle shape, the base sequence changes a little. That's how it happened, a little, and you can't know that through karyotype analysis. That's right, in the end, one is left and almost dies. It occurs then. There's something like this. The nerves are broken, and it doesn't work. So, like a balloon moving around. It's called Huntington, when one expresses it like this. Huntington, all bones. Like this, there are 5, of course, color blindness and many other things. But it doesn't come out well. This, which cannot be known through karyotype analysis. Don't ask in detail what disease it is, because as you may know, there's a lot to reveal, and so on. This is what disease. It doesn't happen like this. Instead, can it be known through karyotype analysis? It's interpreted as whether it can or cannot. That's how it was. Red blood cell karyotype. Can it be known through analysis? Can't do karyotype analysis from red blood cells. So, in the end, it has to be there when first made. Mature red blood cells already have degraded nuclei, so there's nothing to do. Because when solving the test questions, such life sciences are actually in the front. The problem is how much knowledge you have. That's the key to life science. If you don't know from the viewpoint of memory, you get it wrong. I have calculation problems. No matter how hard you solve it, if you miss it from the front, it's over. So at the front, you shouldn't casually mess it up. You have to know it perfectly, what and how it happens perfectly. And it's summarized like this. Guys, take a look at the workbook. Now, I will turn the page and explain in this flow. Now, let's look at it. We will write everything down. Let's delve into it in detail. Our textbook starts with chromosomal numerical abnormalities. So if we go back to chromosomal numerical abnormalities, once again, let's take a look. Once again, I will skip the part where the number of chromosomes is less than normal. Now, chromosomes. I will summarize the numerical abnormalities once again. There are things expressed in detail there, right? It's not a big deal if you hear about chromosomal numerical abnormalities. I keep taking notes though. Chromosome, number of chromosomes, chromosomal numerical abnormalities, mutations. Here, the teacher will divide into teams a bit. There were sex chromosomes earlier. Before that, if we divide once more, we have polyploidy. It's nothing special, aneuploidy. We divided aneuploidy here by sex chromosomes. Polyploidy, guys. Originally, we have 2N chromosomes, right? 3NN. There are things like seedless watermelons at 4N, right? That's a polyploidy mutation. Aneuploidy is just 1 or 2 different things. So we are doing the 1 or 2 different things now. The number of certain chromosomes is 1 or 2 more than normal. You know what this 1 or 2 means, right? 1 or 2 different, such expressions. So the number of autosomes is already more than before. It's already been noted. What should I say? What should I write? An abnormal number of sex chromosomes is due to non-disjunction of autosomes. Fill in the bracket space. Fill in the bracket space. Why is an abnormal number of autosomes due to non-disjunction of autosomes? Non-disjunction occurred in autosomes. Autosome non-disjunction. Next, an abnormal number of sex chromosomes is obviously sex chromosomes. Non-disjunction happened. Got it. Fill in Down syndrome. What is the chromosomal composition? How should I fill this part? Fill it in once again. You have to write both female and male, of course, 45 plus XX 45 plus XY. Edwards syndrome is the same, just like that. Only the numbers are wrong. 45 plus XX. Fill it in on your own when you look again. Done. I filled in Turner syndrome. How? 44 plus X5. I filled it in. Next, because it's not you guys, the teacher will just pass by while only filling in the bottom of the first meiotic non-disjunction. I will fill it in slightly, just once. What happened with the first meiotic non-disjunction that you studied? Guys, you know, right? I'm not going to do any more, but I'll write this as I go. Just check if what you wrote matches with mine. You know, M+1, M+1, N1, N1. I'm just doing this to add to the notes. So for example, when a normal man undergoes non-disjunction, I'll just add this. Always, when XY undergoes non-disjunction first, it goes like XY, XY, 0. If this is fertilized with a normal ovum, I won't write it twice. Now, how can Klinefelter's syndrome or Turner's syndrome be born? Here, guys, there are no super females or super males. They are not born here. I'll just go with this, since you all know the numbers. Just like meiosis 2, I'll reduce the explanation for it. Since we already know about mitosis 2, I'll keep it simple. When non-disjunction occurs in meiosis 2, as the teacher said, since we've already finished studying, so we saw earlier in the problem, it goes n plus 1, n1, nn. When a normal man undergoes non-disjunction, how XX doesn't go in, YY is pushed out, or YY goes in properly. Then, it doesn't go in. It doesn't go in, and there's no one who doesn't know the normal. If this is fertilized with a normal ovum, in one go, a super female can be born. A super male is born, Turner's syndrome can happen, okay? Turner's. And Klinefelter's syndrome is not here. A man underwent non-disjunction, but Klinefelter's syndrome was born in this division. Then it's okay to check it as wrong, remember that. But you don't really need to memorize it, you can interpret it. So that's the end. Then, next to it, I think you can just read the polyploidy, what, 3n? Originally, it was 2n. The number's weird, so it's 3 and 4n, like this. Seedless watermelons are made on purpose. So seedless watermelon is a triploid. Tomatoes are tetraploid. Wheat is hexaploid. There are such polyploidies. This is a mutation. All species should end with 2n. It's a mutation. So now we're learning something new. I'll go back to this. This is chromosomal structural abnormalities. We're done with the number. But now, since I know this, it's visible. Why was it so simple?